Okay, so here you can see the full double-stranded DNA molecule. This is how it actually looks in cells. Let's start off by looking at the sugar phosphate backbone. So in the double-stranded molecule, we have one sugar phosphate backbone here, and we have another sugar phosphate backbone here. And these are actually running in opposite directions. And we use the term anti-parallel. So we say that the two strands of DNA are anti-parallel. We can also see that the 5' and 3' ends are opposite on the two strands. So those numbers refer to the carbons of the sugar. If you go back and remember our labeling of the carbons of the sugar, so this was carbon 1, carbon 2, 3, 4, and 5. So this is called the 3' end because the number 3 carbon is not attached to another nucleotide. And then if you go up here, so this is number 1, 2, 3, 4, and 5. This carbon is not attached to another nucleotide. It is attached to the phosphate group, but there's not another nucleotide attached here. So we call this the 5' end. So you can see that the ends are actually opposite. So we say that these strands are running in opposite directions and they're anti-parallel. And then into the middle, we had the bases. Okay, so you can see that on both strands, the bases are pointing toward the middle and they're paired up and they are bonded with a type of weak bond called a hydrogen bond. And that's what these little dots here indicate. They're also paired up in a certain way. C, which is cytosine, always pairs up with G, which is guanine, and T, which is thymine, always pairs up with A, which is adenine. Now, if these strands were in an actual cell and making up a chromosome, they would be much, much longer. So this is just a small snapshot. And also they would be twisted into what's called a helix. And you'll see that in another picture.